The usual story of fertilization is that millions of sperm race towards the egg, and one sperm, usually imagined as the fastest or strongest, wins. But a series of scientific papers published over the last few years suggest the story is not that simple. These 3 studies found that the female reproductive system can influence which sperm are more likely to succeed, and that this process appears to be linked to compatibility between the immune-related genes of the male and female. In other words, fertilization may not simply be a competition between sperm. It may also involve a hidden compatibility test taking place at the level of the gametes themselves. And perhaps most surprisingly, the sperm favored at the gamete level are not always associated with the males that females find most attractive. So the question is, how can an egg or the female reproductive tract influence which sperm succeed? And why would evolution produce such a system? The traditional sperm race model gives sperm most of the agency. The sperm compete, the egg waits, and the fastest sperm wins. But in reality, sperm move through a chemically and immunologically active reproductive environment. Sperm have to pass through cervical mucus, survive the female reproductive tract, undergo activation, and respond to chemical cues around the egg. Sperm activation includes several processes: capacitation, hyperactivation, chemotaxis, and acrosome reaction. So sperm are not simply swimming in a straight race. They are being physiologically modified by the female reproductive tract as they go along. 2 fluids are especially important here. Cervical mucus, the mucus sperm must pass through earlier in the reproductive tract, and follicular fluid, which is the egg-associated fluid released around ovulation. Follicular fluid contains chemical signals that can affect sperm behavior. Sperm can detect chemical ligands through molecular receptors on their surface, and these signals can enhance or hinder sperm motility. This raises a key question. Does the female reproductive tract attract sperm in a general way, or does it favor sperm from some males more than others? That is where the paper by Fitzpatrick et al. 2020 becomes important. Around 32% of couples undergoing fertility treatment in the UK have no identifiable cause for their infertility. This is known as unexplained infertility. Fitzpatrick et al. Proposed that one possible explanation may lie at the level of the gametes themselves. Specifically, they investigated whether chemical signals associated with the egg could favor sperm from some males more than others. This idea is known as gamete-mediated mate choice, or cryptic female choice. The hypothesis is that chemoattractants released around the egg may bias which sperm are encouraged to approach and potentially fertilize it. This figure shows how researchers tested that idea. They collected sperm from 2 males, follicular fluid from 2 females, and the sperm were placed into a petri dish with thin microcapillary tubes containing follicular fluid. To enter a tube, the sperm first had to swim across the dish, locate the tube opening at the opposite end, and then swim the entire length down the tube. The researchers could then count how many sperm successfully accumulated inside each tube using a microscope. This means that sperm accumulation served as a proxy for how strongly the sperm responded to the chemical signals presented in the follicular fluid. The clever part of the experiment was that every possible male-female combination was tested. Female 1 with male 1, female 1 with male 2, female 2 with male 1, female 2 with male 2. The researchers then performed 2 versions of the experiment. In a simultaneous design, sperm were presented with follicular fluid from 2 different females at the same time. In the non-simultaneous design, sperm were presented with follicular fluid from a single female and a control medium. This allowed the researchers to ask whether sperm preferentially accumulated in certain follicular fluids and whether that depended on the specific male-female combination. This distinction is important because there are three possible explanations. Some females may produce follicular fluid that is generally more attractive to sperm. Some males may produce sperm that are generally more responsive. Or sperm responses may depend on the specific male-female interaction. And this third explanation is the important one. If sperm from the same male respond differently to follicular fluid from different females, and the same follicular fluid attracts sperm from some males more strongly than others, then this is not simply a matter of sperm quality, this is compatibility-dependent sperm response. And that is exactly what Fitzpatrick et al. Found. Sperm accumulation was significantly influenced by the interaction between male and female identity, meaning that follicular fluid did not affect all sperm equally. Some male-female combinations produce stronger sperm responses than others. This provides evidence that egg-associated chemical signals can mediate cryptic female choice at the gamete level. However, the study does not tell us what compatibility signal is actually being detected. It establishes the phenomenon. So the next question is mechanistic. What biological traits make one sperm-female combination more compatible than another? This is where immune genes become important. Magris et al. 2021 is the most mechanistically interesting paper in this story. It suggests that sperm viability in female reproductive tract secretions is associated with structural dissimilarity in immune-related genes. Before looking at the figures, the immunology needs to be clear. MHC means major histocompatibility complex. MHC is the general term across vertebrates, but in humans the major histocompatibility complex is called the HLA system. HLA stands for human leukocyte antigen. HLA genes encode antigen-presenting molecules. These molecules sit on cell surfaces and present antigen peptide fragments to T cells. An antigen is the thing being recognized by the immune system, often a protein from a pathogen broken down into a peptide fragment. It can also be a tumor protein or foreign tissue. So the HLA molecule is not usually the antigen itself, it holds and presents the antigen to a T cell. Think of it like this. The HLA molecule is like the display stand. The peptide antigen is the item being displayed, and the T cell receptor is the inspector evaluating the item. Essentially, the HLA molecule takes the antigen peptides within the cell it's attached to, presents it to the T cell, and asks if this looks okay. If it does look okay, and does not appear to be a threat, the T cell will leave it alone. But if it is determined to be a foreign pathogen, then the T cell will begin to dismantle the cell and any others carrying that pathogen. The T cell examines the peptide and the HLA molecule together as one complex. Different classes of HLA genes encode different HLA molecules, and there are a few different types. But Magris et al. Were specifically interested in allele variants in the MHC class I genes. An allele is a different version of the same gene. You inherit one allele from your mother and one from your father. So for every gene, there are always two alleles. And small differences between alleles can lead to differences in the proteins they encode. So the researchers first genotyped the male and female participants to infer which HLA alleles they carried. They could then use these allelic differences to infer what proteins they would encode. And from those predicted HLA protein sequences, they used an HLA matching algorithm to estimate which specific surface features would be present on each HLA molecule. Those specific surface features are called eplets. So eplets are these structural patches on the molecule. More precisely, eplets are configurations of amino acids exposed on the HLA protein surface that can be recognized by antibodies. T cells do not usually recognize isolated eplets. Instead, T cell receptors recognize a peptide HLA complex, meaning they examine both the displayed antigen peptide and parts of the HLA molecule together. So the important distinction is this. The allele is the DNA sequence variant. The HLA molecule is the protein encoded by that allele. The eplets are small structural features exposed on the surface of that protein. But importantly, allele differences and eplet differences do not always correlate. Some alleles that appear more similar in their DNA sequence can encode HLA proteins with different amino acids exposed at the surface. Conversely, alleles with more different DNA sequences can encode HLA proteins with similar amino acids exposed at their surface. This is down to how the protein folds, and also is due to the process of transcription and translation, where some different DNA sequences can produce the same amino acids. This is a complex process that can be covered in another video. But this is why Magris et al. Used the HLA matching algorithm to shift the focus to HLA eplet dissimilarity instead of simply using allele differences. Because the immune system doesn't care what the allele sequence is. It cares about what the surface of the molecules they encode look like. Therefore, comparing eplets was a more functionally meaningful measure of immunological differences. Magris et al. Also looked at immunoglobulin heavy chain dissimilarity. An immunoglobulin is an antibody. Antibodies can recognize eplets directly. Antibodies are Y-shaped proteins that act kind of like the body's security guards. Their role is to identify and bind to antigens. Antibodies have two heavy chains, two light chains, a variable region which is the antigen binding part, and a constant region which determines the antibody class and how it interacts with other immune components. The heavy chain type helps determine the antibody class, such as IgG, IgA, IgM, IgE, or IgD. When Magris et al. Talk about immunoglobulin heavy chain dissimilarity, they are referring to genetic and structural differences in the immunoglobulin heavy chain region between the male and female participants. Now, the key finding. Magris et al. Exposed sperm to female reproductive secretions and measured sperm viability. They used both follicular fluid and cervical mucus, and they tested whether sperm viability was associated with HLA eplet dissimilarity and immunoglobulin heavy chain dissimilarity. As you can see here, where the eplet dissimilarity was greater, the sperm viability was also greater, and where the eplets were more similar, sperm viability was lower. This panel A, this is the follicular fluid, and then panel B here is in the cervical mucus. As you can see, it's not quite the same relationship, and it turned out that it wasn't a significant relationship here. And here, this is with the immunoglobulin heavy chains. In the follicular fluid, it's a little bit all over the place. There's no significant relationship here. But in the cervical mucus, there was quite a strong relationship where the greater the immunoglobulin heavy chain dissimilarity, the greater the sperm viability. So in other words, the Magris paper suggests two access steps where the right sperm are encouraged to proceed through the reproductive tract. First, in cervical mucus, sperm viability is enhanced when male and female immunoglobulin heavy chain structural dissimilarity is higher. Second, in follicular fluid, Sperm viability is enhanced when male and female HLA eplet dissimilarity is higher. This gives progressive phases of immunological filtering. Earlier in the reproductive tract, cervical mucus favors sperm associated with greater IgH, genetic/protein structural dissimilarity. Later, closer to the egg, follicular fluid favors sperm associated with greater dissimilarity in HLA molecule surface features. But there is an important caveat. The cervical mucus and follicular fluid are not directly reading the male's DNA. They cannot detect an allele inside the sperm nucleus. They can only interact with molecules on or around the sperm cell. So when the paper says sperm viability is associated with HLA eplet dissimilarity or IgH dissimilarity, this does not necessarily mean that the fluid is directly detecting those exact molecules. A more cautious interpretation is this. HLA eplet and IgH dissimilarity predict sperm viability, but the actual molecular signal being detected may be an indirect proxy. The HLA or IgH genotype may correlate with sperm membrane proteins, glycoproteins, glycans, or other molecular features on the surface of the sperm cell. The female reproductive fluid may interact with those sperm surface features. So the genetic differences are not necessarily being detected directly. They may be markers of deeper biochemical compatibility between sperm and female reproductive environment. This is why the Magris paper is so interesting. It suggests that cryptic female choice may involve immune-related compatibility, but the precise molecular mechanism is still unresolved. Jokiniemi et al. 2025 adds the most surprising layer. This paper asked whether pre-mating odor preference aligns with post-mating sperm performance in follicular fluid. This is because it has been found that the types of major histocompatibility genes, MHC, which remember are also called human leukocyte antigen, HLA in humans, are strongly associated with the perceived attractiveness of body odor across multiple species, including humans. The study used 10 females and 11 males. The females rated male body odors. Then the researchers measured sperm motility in follicular fluid from those same females. They also compared HLA similarity between each male-female combination. So as you can see here, in the Jokiniemi paper, the odor pleasantness strongly correlates with the HLA allelic similarity. So the more similar the alleles are that encode the HLA, the more pleasant the odor was rated by the women. And then sperm motility across 60 minutes. There's a weak negative relationship here where the more similar the HLA alleles, the less motile the sperm were. And again, with odor pleasantness, because it correlates with HLA allele similarity, it's the same relationship again. So the higher the odor pleasantness was rated, the lower the sperm motility was across 300 minutes. So this 2025 paper suggests odor acts as a proxy for immunity-related genetic similarity. And at the pre-mating odor level, females rated HLA-similar males as more pleasant, meaning that females preferred the odor of males that were more genetically similar to them. But at the post-mating gamete level, HLA dissimilar combinations showed higher sperm motility in follicular fluid, meaning that sperm with dissimilar immunity genes were encouraged to proceed and swim more strongly through the female's reproductive tract. This is where it seems counterintuitive. If females prefer genetically similar males at the pre-mating level, Then why does their egg and reproductive tract prefer genetically dissimilar males at the post-mating level? The women in the Jokiniemi study were relatively older, with an average age of 35, whereas other studies that have been conducted in the past used younger female study participants. Jokiniemi themselves proposed that there could be age-dependent changes in female mating preferences, and that previous studies conducted with younger females found that young women preferred the odor of genetically dissimilar males at the pre-mating level. This could be because younger females show more exploratory behavior, seeking more dissimilar male partners, which in our species' ancient past would have been equivalent to finding a male outside of the tribe, whereas older females could be more likely to seek guaranteed safety and security by finding a male within the same tribe. But regardless of the pre-mating preferences dependent on the life stage of the woman, the gamete-level sperm-egg compatibility has the final say. Taken together, these three papers suggest that fertilization may involve more than simply selecting the fastest sperm. Fitzpatrick et al. Showed that follicular fluid can differentially influence sperm behavior depending on the specific male-female combination. Magris et al. Showed that sperm viability in female reproductive fluids is associated with immunological dissimilarity, specifically HLA eplet dissimilarity in follicular fluid and immunoglobulin heavy chain dissimilarity in cervical mucus. And Jokiniemi et al. Showed that gamete-level compatibility may not always align with pre-mating attraction, with follicular fluid favoring HLA dissimilar combinations even when odor preference favored HLA similarity. The obvious question is why? Why would the female reproductive tract favor sperm associated with greater HLA and IgH dissimilarity? A very likely possibility is that this increases immunocompetence in the offspring. HLA genes encode antigen-presenting molecules that determine which pathogen-derived peptides can be shown to T cells. Offspring inheriting a more diverse set of HLA variants would therefore be capable of recognizing and responding to a broader range of pathogens. Similarly, variation in immunoglobulin-related genes may contribute to greater diversity in antibody responses. In a world where infectious disease was historically one of the largest causes of infant mortality, mechanisms that increase immunocompetence in offspring could provide a significant evolutionary advantage. Another possibility is inbreeding avoidance. Close relatives tend to share many immune gene variants. If reproductive fluids preferentially favor sperm associated with greater immunological dissimilarity, this could reduce the likelihood of fertilization between genetically over-similar individuals. Importantly, the reproductive tract would not need to recognize relatives directly. It would only need to favor molecular signatures associated with genetic dissimilarity. This could provide an elegant biological mechanism for reducing the risks associated with inbreeding. Both of these hypotheses make evolutionary sense. For most of human history, infant mortality rates were extremely high. Infectious disease was common, and mating between genetically related individuals was much more likely than it is in large modern populations. Under those conditions, mechanisms that improve offspring immune function or reduce inbreeding could be strongly favored by natural selection. However, there is an important caveat. Greater dissimilarity is not necessarily always better. There may be an optimum rather than a maximum. Extremely high immune gene diversity may also carry costs. Potential trade-offs include T-cell repertoire dilution, where increasing the number of HLA variants can complicate immune recognition and T-cell selection. increased risk of certain autoimmune conditions associated with particular HLA combinations, and pregnancy-related immune interactions, where maternal and fetal immune systems must successfully tolerate one another throughout development. So the goal may not be to maximize difference. Instead, the reproductive system may be selecting for an optimal level of immunological compatibility. Not too similar, not too different, just different enough to provide the greatest overall reproductive advantage. And if that interpretation is correct, then fertilization is not simply a race between sperm. It is a compatibility assessment occurring at the molecular level. A process in which the female reproductive tract may be evaluating biological information that neither partner is consciously aware of. And that may be why the beginning of a human life depends on much more than simply which sperm reaches the egg first. Thank you for watching. This is Nexum, a new channel I'm just starting up again, where I explore emergence of complex systems and intelligence. Specifically, I'm going to be covering a lot of biology and computers, but also philosophy, any new scientific research that comes out, and any interesting theories. I'm a computational biology master's graduate, and I figured that I'll start making videos on all the content that I find interesting. This is the second video I've ever made, and I'm still getting the hang of it. But if you like this video or you learned something new, please like and subscribe. That will really help me out on the algorithm, and I'll be able to keep creating more videos.